for lesson 2 group problems. Tanner and Roxana, what's the name of the other person you invited?
Sarah.
Sarah?
Mhm.
Okay. So Tanner and Sarah are not present right now. But they would normally be here. Just disclosing this for homework purposes. Don't even know if that's necessary. So why don't we just kind of go through the questions and talk about how we got there and just kind of cross-check with people to make sure that we all have the answers. Oh, hi, Sarah. Welcome.
Hi. Oh my gosh, it's pink. I'm sorry.
Oh, it's okay.
Hi.
Oh, geez.
So I don't know if Tanner is coming, but this is a pretty good group now. So
What?
oh, so we would have one other person probably, but I don't know where he is right now. But I sent him the invite. But I think we're just going to go ahead. So Sarah, I'm just going to fill you in on kind of what the plan is. Up here, I have the group. Yeah, these are the group problems for lesson 2.
Okay.
And I had already turned mine in and I corrected it with the answer key that became available. So these should all be the correct answers. We're going to go through them and talk about how we arrived at the problems.
Okay.
And kind of just make sure that everybody's understanding is catching up. Does that sound good?
Yeah. Sure.
All right. So the first problem is about blood types with humans and how ABO is an independent trait from the rhesus trait, and both of those are also independent from the MN blood trait as well. So problem A talks about a child with type O, Rh negative, and M.
Mm-hmm.
And so he is born to a woman who has the blood types O, Rh negative, and Mn, and a man with A, Rh positive, and M. So we are supposed to determine possible genotypes for each parent for this child. So this is the only possible genotype for the child because
The information.
O is recessive, Rh negative is recessive, and because M and N are codominant, the only way you can have M is if you're homozygous for M.
Mm-hmm.
Perfect.
Okay, and so then does the following for mom and dad look right to you guys? And do you guys kind of understand how we would arrive to those answers?
Yeah, I got the same answer. Yep.
Okay.
Perfect. Yep, me too.
So what about you, Sarah? Does mom and dad look right to you?
Mm-hmm.
Okay.
So just to make sure, so this is what I did for that. I don't know if you guys can see it. But I basically broke it down, and then at the end, I was able to figure out the genotype and all of that.
Yeah, that's pretty much how I did it as well.
Yeah, just okay, cool.
All right, so then part B, we're to find the proportion of children born to a man with this genotype and a woman with this genotype. We're supposed to figure out the proportion of children who will have B, Rh negative, and MN. So that means the possible genotypes for the kids, or at least for B, could be I to the B, so IB or IB little I.
Uh-huh.
And for Rh negative, you have to have RR. For MN, and for MN, you have to have MN. So, did everybody come to this?
Yep.
Yes.
Okay. So yeah, just stop me if this feels like it's going fast or at a breakneck pace, or we need to talk about something or explain it.
Yeah.
All right.
That's the same. Sweet.
So then part C. What proportion of children are will have A, RH plus, and M when they're born to a man with this genotype and a woman with this genotype? This is the answer that I got. Do we all kind of know how to arrive to these proportions?
Yeah, I got the same one.
Okay.
I did it wrong, but I understand why I did it wrong.
Okay.
Because I said RH negative,
Okay.
RH positive. So I got the same as B, but then I was like, oh, wait. So yeah, that makes more sense.
Okay. Sarah, does this make sense how we arrived for this?
If I have a question, if like not understanding something and like how you guys got there, I will ask you. Don't have to worry about.
All right. Sounds good. All right. So then we'll just keep moving along then. Okay. So a man with the blood type B, RH plus, and N says he cannot be the father of a child with blood types O, RH negative, and MN. The mother of the child has a RH positive and MN. Is this man correct? I said he's incorrect because it is possible for it to be his child. I kind of went into a lot of explanation more so than what the teacher did on the answer key.
I got the same though. 1/32. Yeah. Yep.
Yep. For the possibility or proportion of options.
Yeah. So even though it's small, there is still a high probability.
Yeah.
Okay. So then this one. In class, we learned that the shape of that the comb shape of chickens is determined by two alleles, R and P, and these are the different types of alleles. Talks about how walnut is produced when both genes involved contain at least one dominant allele. So capital R blank, capital P blank. Rose comb is produced with at least one R allele is present, one capital R allele is present, so it'd be capital R blank, little p, little p.
Mm-hmm.
And the P comb is produced when the first gene contains two recessive alleles, while the second gene contains at least one capital P allele, so little r, capital P blank. And if an individual is homozygous recessive for both, there is a single comb. Determine the genotypes of the parents that produce the following types of ratios. So this one I initially got wrong, but I understood where I got that wrong once I worked it through.
Wait, so letter A, sorry.
This is the right answer, but I'm just letting you know that this is one of the ones that I got wrong initially.
Cool. Letter B, I got it wrong. I have big R, big P, little p, and big R, little p, little p. Why? Hold on.
So, if you guys have questions about any of these answers, just holler out, and I will talk about it. So
that's weird, because when I was working through letter B, I don't know if you're able to see my work, but basically I got the same proportion, but with different parents. But maybe I did it wrong. I don't know.
These ones are kind of a bit tricky. How I worked it out is I basically set up a Punnett square. I don't know how I would do this on the computer.
Right.
Let me try something out real quick.
Write it down.
Yeah, let's do this. Okay, so here's how I kind of set it up when I was working through it. So we have 3 rows, right? So rows, you have to have big R, then there's a blank, then you have to have little p, little p.
Yeah.
You have 3 of them, big R, blank, little p, big R, blank, or nope, that one should not go there. Big R, blank, little p.
How do you know where you place it?
It doesn't really matter where you put them. This is just how I personally set it up.
Oh, okay.
And so then I kind of worked backwards, where see what's common right here is that there's an R right here, is that there's a capital R in both of these rows, and that there's a little p in both of these rows. So that means that this gamete would have to have
Big
R
R.
and little p. And then this one, there isn't a common R, so we'll worry about that one later.
But the P,
But the
little
P
P.
is the same. So you have to have a little p. And so then we also know that this one, the only other option for here,
Big R.
little p.
Wait.
So we'll worry about the big R later, but
Okay.
yes. And then also the same over here,
Little
little p. However, because this has to receive a little r from both parents, that means a little r is donated from here and here. Does that make sense?
Yeah.
In order to make the gamete. So then I filled that in. And so now you can have RP, and then you can get this one, which is essential. And so then I plugged in, well then, right here, there has to be a little r. And then right here, there has to be a little r. And so then where did I put my notes?
So that means that the P, the little P app, the one that is on the top, that would have a big R, right?
Yes, because we determined that this one had to donate one of the little r's over here. This means that this big R comes from up here.
Right. Oh, okay.
Does that make sense?
Yeah.
Not really.
So not really for who? What? Okay, Sarah. What did not? How did you? Or where did you get confused?
Like, are you doing like, I think I confused, like, this is the pan square, and then you just like dividing? Not dividing, but this the, as I said, like mom side and dad side?
Mm-hmm.
Okay, so, no. So I'm fine. I'm sorry.
No, it's okay. So I'll just put these in bold so that way you can know that these are like the parents. And they only have 2 right here because I don't know why I did it like this, but it just made sense in my brain on how to compute it. And that's pretty much how I went about doing all of these, except for this one. This one was a little bit
Yeah, I don't think I get that one, but we'll worry about that later.
yeah, this one and this one. It was this one and this one were just a little bit more complicated.
So you basically just, based on the phenotypes that you were given, you broke it down into, okay, what are the possibilities for the genotypes that would give me this phenotype? And
Yeah.
then you just did the Punnett square, and based on the phenotypes, you figured out the parents.
Yes.
Pretty much. Okay. Yeah, that's really smart. I like that. Cool.
So yeah, that's how I did this one and this one. This one was a bit different because I'll just kind of talk through how I got this one. So you remember this one was actually one of the ones that I got wrong. Because what was walnut again? Sorry.
Walnut was big R space big P space.
Yeah, where they had to have at least one of them.
Uh-huh.
So,
Yeah, I was like, oh, I couldn't even, like, I gave up, honestly. I was like, yep, I don't know. I need to ask
this one was one of the ones I got wrong initially as well, but then when I checked it with the answers, I got this same I got the same ratio that they ask for right here. So as far as working backwards for this one, I was a little bit more at a loss. It was a little bit trickier.
Gotcha.
So for this one, I worked out the cross and the probabilities for each of the possible genotypes, and it gave me the correct ratios for these phenotypes, but I just wasn't able to figure out how to work this one backwards.
Yeah, okay. So you have to do it like little step by step kind of thing.
Yeah.
That's how I was doing all of them. I was trying to kind of like figuring out based on the phenotype what are the two possibilities of genotypes, and then I would try to like set up the Punnett squares and kind of like work kind of like back and forth, trial and error pretty much.
Yeah, at this
Yeah.
point it kind of has to be a trial and error
Yeah.
to some degree. Um
But no, yeah, really. C and D, I was like, yeah, I was so confused because I tried and tried and tried but I like the way that you set it up though. And probably it's not gonna work for all of them, but probably for most of them it will.
yeah, for most of them it will. So that's the nice thing about that.
Okay.
Sorry, I'm just kind of looking at this and trying to work it backwards in my notes just to see if I can get the same genotype because I realized I did something wrong.
I feel like there's more than one right answer on this one.
I feel like there could be more than one right answer on this one as well because when I did purely heterozygote cross, I got 9/16 for walnut and 3/16 for rose, which is the same ratio as 3 walnut to 1 rose. But there are more possible phenotypes you could get, but it got you the same cross. So I feel like some of these could have multiple answers.
Yeah, 'cause, like for letter B, was it? Yeah, for letter B, I got a different, not for letter B or for letter A or C. Well, one of those, I can't find it right now. I got a different answer, but the proportions were correct, and I checked them like 3 times to make sure. And so I think it was for C. Yeah, for letter C, I had a different answer. And I still got the proportions right.
Yeah. And I feel like you could even put a big R right here and still get the right answer.
Uh-huh.
Like it doesn't have to be lowercase.
Yeah.
Because it says at least one dominant allele. So yeah, this one and this one, I feel like there could be more than just one right answer.
What was B? Let's see. But anyways, yeah.
But so that's kind of how we worked through those ones. So I don't want to do this one because that's just extra time.
Yeah, very cool. I think it's cool.
Okay, so this one was about horses. Palomino horses have a golden yellow coat. Chestnut horses have a brown coat, and Cremello horses have an almost white coat. So here are the different coat color crosses and the ratios of things. And
I have a question. How, because I'm having problem figuring out ratios. How do I figure out ratios? Because at top it's just like not the Penn square. I thought I would just use that thing and then multiply the result.
Explain one more time so we're back up here.
No. It's like ratios in general. Because I
Oh,
thought, like, every time I tried to get the ratio, I think I got the proportion right, but every time that I tried to get the ratio, I got the ratio wrong, and I don't know why I got the ratio wrong. So I don't know if, like, because I
Well,
thought
it depends
multiply.
Right,
what
but
you're
the
trying to
ratio
figure out. So, for example, if you're working with genotypes, you have to multiply the numbers. But if you're doing phenotypes, you have to figure out how many genotypes give you those phenotypes and then add them. So depending on
Right.
what you're looking for.
So
Yeah.
it's like, so if you have, didn't get it. So genotypes, I got this like multiplication. So we don't care about color, don't care about anything. Just care about the alleles, that one.
Yeah.
Yeah.
So here's an example. I'll just put it in green so that way we can differentiate. So let's just work with, I'm going to highlight this one. Let's just work with this cross for right now and just talk about a palomino x a palomino. And here's our genotype for a palomino cross right down here.
Mm-hmm.
So I'm going to put in another 3x3 table. So we'll do a palomino cross. So here's palomino 1. So big C and then little c. And here's big C and then little c. So then you get capital C, then you get capital C, little c, and then little c, little c. So that means we'll just work with genotype right now. So that means 1/4 is big C, big C.
Mm-hmm.
And then we have
1/2 is little c.
So
No,
1/2,
little c.
1/2 is big C, little c.
Mm-hmm.
And then 1/4 is little c, little c.
Yep.
However, let's say that this is not incomplete dominance. Let's say that this is full-on dominance where the chestnut takes over. Where if you have at least one of these big C's, it's chestnut.
Mm-hmm.
So we'll just take out palomino right now. We'll just say palomino is a different name for heterozygous right now. So phenotype then, if this was complete dominance, which is different than what is shown here,
Mm-hmm.
if it was complete dominance, then we would have actually 1, 2, 3 that would show the chestnut color.
Yeah.
So 3 that would show chestnut,
And 1.
and then 1 that shows cremello.
Honestly, for this problem, I thought that it talked about the what is it called? The polygenic inheritance, where you add the dominant alleles. But then I realized that that was wrong. So
Yeah. So we'll talk about that here in a second. I'm just going to explain how you kind of get this ratio from these. Let me actually zoom in on this. This will help everybody out just a bit more. There we go. That'll make this a lot easier. Okay, my bad. So just working with pure numbers right now. So we have 1/4, big C.
Mm-hmm.
But then we want to add the 1/2 big C, little c, because we're assuming that this is also dominant in this case. So you can't add 1/2 to 1/4 just because they don't have the same denominator. So then what we'll do is we'll change that to 1/4 big C plus. So we need to get this to the same denominator. So we'll multiply it by 2. So that way 4 is at the bottom. So that means 2/4 big C, little c. So now we can add it, and that means we have 3/4 dominant phenotype. Does that make more sense? how
Yes.
we're going to get this ratio. And so
Yes.
see, we have this 3 right here and this 1 right here. So that means we still have this correct ratio.
Yep.
Okay.
Whereas now, if we were to just say, whoa, if we were to now consider that this is incomplete dominance, we'll say that there's
1,
one chestnut,
2, Palomino.
2 palomino. I can't spell mellow. So this is different because it's incomplete dominance. This means that the big C and the little c like to find some happy medium between each other.
Mm-hmm.
So this is where we get our different ratio. However, the math will still hold because remember we have to multiply this fraction by 2 to get the same denominator. So you would still have 1/4 big C, then you would have 2/4 big C, little c, and then 1/4 little c. So the numbers still hold up.
Okay.
Does that make sense?
It does make sense. Thank you so much.
Yeah, so I hope that kind of helps. And it kind of answers this answer up here for why it's incomplete dominance because they share the traits. So everybody on the same page with that?
Yes.
Couldn't it also be codominance though?
Codominance is when it would actually display both traits. So codominance would be back up here with ABO blood type, where
Yeah.
where IA and IV are both dominant, and so you can display AB blood type.
Yeah, because codominance it displays at the same time.
Yeah.
And it's not really blended. But with coat color, it can appear blended when it's really not.
So with coat color, Palomino really is a blend of these two colors because this is kind of like a dark brown, this is a white, this is like a yellow. So it's like an in-between. So it's like you mix a red flower with a white flower.
You get white flower. You
flower.
get.
Whereas, like, if so,
It's not
where you,
passing.
yeah, it's not displaying both brown and white at the same time. Does that make sense?
Yes. I just I don't pay attention to horses and colors. And it said like the golden brown, but I was like, does that mean like golden brown like patches with like white but yeah, okay.
No, it just means like a yellow coat color. And I only know that because my sister was big into horses when she was like 5. So yeah.
So quick question before we move on. For number 3, do you guys remember talking about on the slides about polygenic inheritance and how if you have 3, you could have like a gene or several genes that code for the same trait, and if for each gene you have the dominant allele, they kind of add, which increases the color pigmentation of the skin or whatever. And so I thought that had to do with this one. And my question is it because we're talking about horses and not the skin pigmentation of, I can't remember what the example we were given in the slides, but the other mammal?
So you're talking about kind of like the rabbits. So that's where like so that's different. This is incomplete dominance. This is where you still only have like 2 different alleles, but the heterozygote has a blend. Whereas polygenic inheritance will have let's say
like different genes that code for the same trait, but they're
it'll be different alleles on the same gene.
So
This is what you're thinking of. So this is like on this gene you can have let me put in,
and all of those alleles code for color.
They'll code for different colors, correct?
Right.
So I'm just going to put in an example right here so you can have like, I'm just going to use the example of rabbit hair color that they have. So C means a full coat. Where C to the CH means a chinchilla coat. or C to the H means a Himalayan coat, or like it'll be black around their noses and limbs because their body can't produce the white pigment at their extremities. And then you'll have little c. See how you have these 4 different alleles, whereas here you still only have 2 different alleles?
Yeah.
This is polygenetic inheritance. This is where you have like
That's actually multiple alleles.
or yeah, this multiple alleles. Sorry, thank you for
Yeah.
that.
Because polygenic inheritance is the skin color, like 3 dominant alleles equals the same skin color no matter what those alleles code for.
Yeah, polygenetic is probably even closer to like this one where you have these 2 genes interacting.
Yeah. So there are several genes that code for the same trait.
Yeah. The R and P genes are coding for this same trait of cone type. Whereas down here we were talking about multiple alleles, and here this is just one gene coding for one thing.
And so my question is like, how do you differentiate between when we're talking about polygenic inheritance versus codominance? Because for a sec, this problem, I thought that it was polygenic inheritance because basically the color is just, the pigmentation of the color is just decreasing. Does that make sense?
Well, the reason why you can kind of see why it's, holy crap, what did I do here? Why it's polygenetic, or not polygenetic, excuse me, incomplete, is that if you look at the Palomino cross, it holds a similar ratio as a regular, as
Mendelian
does, where you'll have, this is nearly 12, and so these are pretty close to 6. You wouldn't expect to see exact ratios in the wild, but this is pretty darn close to 6, and this is pretty darn close to 12. So it's almost 1 to 1. Does that make sense?
Yeah. So basically, the way that you figured it out, it was based on the ratios.
That's how I figured it out, yes.
Gotcha. See, I didn't even pay attention to the ratios too much because I was like, oh, if it's just changing colors, my first thought was like, oh, probably it's a polygenic inheritance where you just add the dominant alleles, giving a darker pigmentation or a lighter pigmentation, but that does make sense.
Yeah.
Thank you.
Okay, so then we'll go to this one. This is just kind of fill in the blank. This one I first got wrong. I thought they were talking about the type of the type of antigen on the cell surface. But then I realized, nope, they were talking about an enzyme, so I had to go back and correct that. So it was
I got that
transferase.
Okay.
And then blood type B adds on another glucose to the acetylgalactosamine.
Okay.
So then I guess I'll just kind of read over the answers and stop me if you got something different or want to talk about it. So B is overdominant / heterozygote advantage. C is incomplete dominance. D is epistasis. E is phenylketonuria or PKU. And and then F is lethal allele. This one is a 60% dextrol to 40% synstrol and 40%.
Question. Okay, so for G, I guess it wouldn't make sense, but how did you get 0.8 or 80% for synestral? Because I got the dextrol one.
40%?
Yeah.
Okay, so I basically just ripped it off of the answer right here. Because it's maternal inheritance, the offspring are going to have the same ratios
Right.
for phenotypes as the mom. So we have 30% show this one, which is the dominant or dextral phenotype, and 30% are heterozygote, but they still present the dextral dominant phenotype. So
Right.
30 plus 30, 60%. So that's why I got 60% dextral.
Uh-huh.
And then the other portion is 40%. So the other 40% would be synstral. And that basically is spelled out here as well, because of that maternal inheritance.
Okay. Oh my gosh, you're so right. I just checked my notes and I completely did that wrong. So never mind.
Okay.
Never mind. Got it. Oh my gosh. Okay.
Yeah, you're fine. Okay, and so then this last one is pretty much the same deal, where this is also female, or this is also maternal inheritance, where 40% of the female plants are green,
Yeah. Got
and then
that
the other
too.
60% are white.
Yep. Got that.
So that's why we'd see it here, because they follow the mom. The genotypes, of course, would be different. However, the phenotype will still be same as mom,
Mm-hmm.
or the phenotype ratios. Anyway, this one was not quite so fun because, well, in the answers, this is one thing, and this one only presents as being this answer, but this only presents as being this answer. However, it is possible for that W to get thrown in there because it would just be overshadowed by these other two. So I just left it up there because honestly, there are two answers for these two. For these two, no. So I'm just giving that little disclaimer right now.
Oh, I see where you're coming from. Okay.
Yeah, sorry, I kind of jumped ahead there. My bad.
No. You're good. It took me a second to catch up. Because
Yeah.
I honestly thought there was a number 5, and so I haven't even worked this problem out.
Okay. Oh, yeah. So this one,
Like on my Word document, it got like pushed to the bottom, and I was like, "Oh, I'm done." It didn't. I wasn't done.
yeah, that wasn't fun. I realized that too, and I almost turned it in, but I was like, no, crap, I got one.
Oh my gosh, no.
It's a struggle.
See, this is where I didn't get to, but I'll promise I'll work through it.
Okay. I could work through this first one with you guys if you wanted.
That'll be awesome.
So it's pretty much similar to question number 3 back there, where we kind of worked through things backwards. So I followed pretty much my same. I followed my same formula because it worked out pretty well, at least for myself. This is the case with multiple alleles, like what you were saying, Natalie, and how I was using it for polygenetic inheritance a while back. So that was my bad. But this is where we have like B, S, C, and W as possible alleles for this one gene. And so black will pretty much override, or so B will pretty much override everything. S will override these two. Cream will override white, and white you have to be homozygous to present. So 50% of the offspring are black, and 50% of the offspring are sepia. So that means that you have to have a B, and then something else here. And then you would, I'm just going to do it like this, B, and then something else here. And then because it's sepia, that means that it has to display this S. at least, but it can't but it can't have a B allele, otherwise it would be black, right?
Mm-hmm.
So that means S here and S here, right?
Yes.
So working backwards, I just kind of did it, or how did I do it in my notes? Yeah, I did it like this.
Mm-hmm. Some big S. Okay.
And so because they're the same, and then you can put S right here, and then it'll be
Big S. Okay.
or like then you can put a C down here, or it could be double C, or it could be SW. Honestly, as long as there's this S and here is one of these other two alleles, you're fine. However, this one has to be black and this one has to be sepia. So this phenotype does not change.
Right. Okay. So basically, you just pretty much what we did before, where you just figure out the genotypes that would give you that phenotype, right?
Yeah, that's pretty much what you do through all of these.
Okay.
And these two on the bottom, I'm just going to let you know, these are the only possible answers. This one, you kind of run into the same deal where you could get like this,
More than one.
or this has to be one of them.
Yes.
Like this CS has to be one of them, but then this could be replaced by either C or W.
W. Yeah. Okay. Got it.
So yeah, you would work through these problems very similarly to how you would work through, or how you worked through up here with number 3. Does that make sense? Kind of how you work backwards, where instead of working with ratios, you're working with percentages, which are just other ratios.
Yeah. I got it. It's really, honestly, I would have never thought about doing that, like going backwards. So that's really helpful.
On the top one, oh, yep, you already put that. Never mind.
All right.
You have SS as well, but you mentioned that.
Yeah.
That's what I would have put, was BS times SS.
Mm-hmm. Which the fact that she only gave one answer, I thought was complete BS. Haha.
Maybe I'll email her and ask her.
I honestly just left that there, but yeah, it would also probably be smart to ask her or like put it in the discussion board.
Yeah.
For this week.
Yeah. I feel like I'm gonna be like that student that just asked her a ton of questions, because I'm like, I just ask a ton of questions.
I have been that student the last week. I'm like, I don't get this. Please help me.
And last week understood better than this week. I'm like, what the heck? It's fine.
Yeah.
Just like,
to be honest, though, I thought that I would hate genetics. I am actually kind of enjoying it because I didn't have a good experience in Bio 181. So I was like kind of a skeptic, but
it's funny. I really liked it in Bio 181, and now I'm like, oh my goodness, what am I getting myself
into? In Bio 181.
At the beginning of the semester, they teach for like 2 weeks genetics.
Yeah.
Oh, ours was in the middle. Well, mine was in the middle.
Or something like that.
In the middle as well.
But yeah. Anyways.
All right. So any other questions about these problems before we sign off?
I think I just need to work through the final one, the last problem, and then I'll probably be good. So thank you for your help. Yep.
Yeah. I have a question.
Okay.
That's just in general, kind of. So this video that we just made, do you just post it, Audrey, and then it counts for all of us, or does it just count for you, and then we have to create our own videos?
Okay, so what's gonna happen
you know?
is that I will take this and because I started it through YouTube, it will just save as a YouTube video, and then I will post it in the discussion board, and I will put your names up on the discussion board because I've done I did a class like this one time online where I had to do the discussion video and then we had to put our names, like reply with our names, but I don't think you have to do that. I if you just on
Like if you just comment on the discussion board, it's gonna be like marked that you did it.
yeah, exactly.
Okay. Cool. Because last week I just recorded my own. So, okay. Cool.
Like
Thank you.
some kind of comments that like just say I was there.
Yeah, you can say
just-
like, smiley face, I was here, whatever, but yeah.
Okay.
Yeah, I would put at least your first and last name though, just so that way it's like if she were doing attendance, it's like, oh, check, check, check,
Uh-huh.
sort of deal. Just make it really easy.
Mm-hmm.
Okay.
All right.
Cool.
Well, then I will see you all around the same time next week on Thursday then.
Sounds
Hopefully
perfect.
closer to 6:15ish.
Sounds good. Actually, that works better for me because I get off lab at 6, and so that gives me time to get here in case lab takes a bit longer, just a few minutes longer. That happened today, so I was like grateful that you pushed it a little bit.
Well,
Yeah.
I'm glad that me being held up at the store helped you out then.
So yeah, that works.
Perfect.
All right. Well, then, if there are no more questions, then I will see you all next week.
Okay, see you guys.
Bye.
Bye.